Good evening, everyone, and welcome to Alumni University. I'm Kristen. I'm the Director of Constituent Engagement here at Illinois Tech. Before we get started this evening, just a few quick housekeeping notes. For participants, your video and audio has been disabled. So if you have any questions, please type them in the chat box at the bottom of the screen. Also, just a reminder, this presentation is recorded and will be available on our website in the next couple of weeks. So to introduce our presenter tonight, Oscar Juárez, Ph.D., is an associate professor of biology here at Illinois Tech. He has both an undergrad and a PhD from the University of Mexico, and his lab here at the university conducts biomedical research directed to establish the metabolic strategies and host pathogen relationships in bacterial pathogenic species. So with that, I will turn it over to Professor Juárez to get us started.
Well, thank you very much for the invitation, Christina, and thank you very much for you guys to show up today. I'm going to be sharing my screen. Let me know if you guys can see it. Can you guys see it?
Yes, we can see that.
Okay, can you see the presentation mode?
Yes.
Perfect. Okay, thank you so much. So in today's presentation, I'm going to be talking more, and this is a little different compared to what my other colleagues have been doing. They have been talking a lot about the things that they're doing in their labs and in the research groups. I'm going to change it a little bit because I think that the message and the mission of the lab is, I think that is the most important thing at this point, okay? So I'm going to be talking to you guys about this crisis that we have with antibiotic resistance. What are the causes of this problem? What are the challenges that we're facing? And what are the solutions that my lab is working on? The first thing that I would like to ask you is, how do you like, how you guys liking the current pandemic? Are we having fun with all of these mandates by the government, by the social distancing, by the mass vaccination, by all of these economic distress that we all have been living through? Are we having fun with that? Are we also having fun? Is anybody having fun in this COVID roller coaster of waves going up and down and up and down and up and down and up and down? And by the way, the roller coaster ride is not over yet. We're still, COVID is here to stay. And since my guess is that most of us are not having fun in this with the current pandemic, now the question is if we are ready for the next pandemic. And then I imagine that many of us were caught off guard by the current pandemic, and we really felt like these sailors in these very famous, now famous Kanagawa wave portrait out in Japan, in the Sea of Japan. Okay. So in this analogy, I can imagine that we're going to have this little nerdy, little friendly antibiotic that we really need in our lives. And this antibiotic is minding its own business, and it is not realizing that this wave of superbugs, of antibiotic-resistant superbugs, is about to crush this antibiotic, and with that also might collapse our health system. So this is a big problem, and I would like to talk to you guys about the problems that we're having with this in this situation. Okay, I'm going to try to make this talk understandable for everybody. So I'm going to go over a few concepts. Okay, so the first concept will be, what are antibiotics? Okay, so antibiotics basically are medicines that we use to fight infections. Okay, these are going to be chemical compounds that we use to kill microbes that produce disease. Okay, and these microbes can be bacteria, can be some other type of organisms, but in general they're microscopic. There is no way in which we can get rid of these microbes unless we use antibiotics. So this should tell you how important antibiotics are for us at this point. Okay. How is it that antibiotics work? How do antibiotics work? Well, basically, antibiotics are going to be inhibitors, okay? These inhibitors are going to be inhibiting certain processes inside of these pathogenic bacteria, of these bacteria that can cause disease. And there are many different types of antibiotics and many different mechanisms of actions, but we can classify them in three general categories. Cell wall synthesis inhibitors, nucleic acid synthesis inhibitors, and protein synthesis inhibitors. By blocking any of these three processes, we will be stopping the growth of bacteria and killing the bacteria. And for that reason, antibiotics work in the clinic, okay? Now, what are going to be the uses of antibiotics? Of course, it is obvious. First of all, we're going to have medical uses, okay? In the clinic, we're going to be using antibiotics to treat infections. For example, many of us, including myself, suffer COVID-related pneumonia. And in these, the only way or the easiest way to clear the COVID-related pneumonia or any type of pneumonia or any type of any other infectious disease, the easiest way to treat it will be through antibiotics, okay? We're also going to be using antibiotics for people whose immune systems are suppressed. For example, during cancer therapy, for HIV patients, after organ transplant, for rheumatoid arthritis, and for lupus, and for many other conditions, we're going to get to see that the immune system shuts down. Our body cannot fight infections, and for that reason, we're going to need antibiotics in these cases. In addition, many of us have had surgical procedures. We're going to need antibiotics to prevent that we develop infections in the area in which that were subjected to these manipulations, okay? After wound treatment, of course, we're going to need antibiotics. In many cases, for those of us who have been subjected to dialysis, for example, people who have diabetes, they do know that it's very common for them to develop urinary tract infections. And in these cases, you will also need antibiotics. And also for certain surgical procedures that your dentist will be carrying out, we might also need antibiotics, okay? So antibiotics, of course, are needed for many different, in many different situations, okay? But also we're gonna need antibiotics for non-medical purposes, okay? So we're gonna need antibiotics, for example, if our pet, and we love pets, okay? I love pets, everybody loves their pets. Anytime that my Tony or my Rocco, those are my two dogs, get sick, we will take them to the doctor, and if they need antibiotics, we'll give them antibiotics, okay? And then antibiotics are also going to be needed for beekeeping, aquaculture. They help in the production of alcohol, for the production of foods, as food preservatives, and we also use them even in soaps, okay? So as you guys can see, antibiotics are going to be pretty much everywhere, okay? We need antibiotics pretty much for everything. But also this has led to the abuse of antibiotics, and I'm going to be talking about that later. So these antibiotics, the use of antibiotics we have access to the antibiotics thanks to the works of two guys, Paul Ehrlich and Alexander Fleming. Both of them are Nobel Prize winners, and one of them synthesized the first synthetic antibiotic to treat syphilis, and Alexander Fleming discovered penicillin. And through these discoveries, into the way in which these guys developed their antibiotics, this led to the explosion of antibiotic discovery, to the golden age of antibiotic discovery. That was about 30 to 40 years, from 1930 to 1970, or from 1940 to 1980s, depending on who you ask. During this time, we really were living the time of antibiotic discovery. As you guys can see, we discovered most of the antibiotics that we are using today. Some of the antibiotics that we use today are derivatives of the ones that were discovered before, but most of the antibiotics that we're using today were discovered during this time. I would also like to notice that after the mid-80s, 1987 and 1988, and certainly after the 90s, for the last 20-something years, we have had a void of antibiotic discovery. And that is a big problem, and that is a big problem that we are trying to address, okay? What is the impact? How can we measure the impact of antibiotics on human health, okay? I understand that I discussed to you guys, I have mentioned to you guys, how is it or why is that on what conditions or what situations we're gonna be needing antibiotics. But how can we measure how important they are? Okay, why don't we, in this plot, why don't I show you the life expectancy of people from 10,000 before the Common Era up to today. Okay, so we're talking about from the beginning of civilization up to, well, this graph is a couple of years old, up to 2020. Okay, during 90% of the time, and our species has been living on this planet for probably 100,000 to 250,000 years. So the situation that I'm about to talk to you guys is probably worse for most of the time in which we have been living in this planet, okay? But at least from 10,000 BC to 1850, most of the people were dying, most of us were dying at 25 years old. We were living very short lives. We were living probably very miserable lives. And these lives were marked by disease, okay? As soon as three factors were introduced into the equation, water cleaning, vaccination, and antibiotics, and after antibiotics in particular, what we could do, what we did as a species is extend our life expectancy. And I think that today the life expectancy of Americans is around 85 years old. And I think that 85 years old is probably a good time to say goodbye, at least to me. But when we extended our life expectancy, we tripled our life expectancy, due in part to antibiotics. Okay, how is it that people? So going back on this situation, okay, what is it? How is it that people were dying 120 years ago? Okay, in 1900 U.S. Let's compare the causes of death in 1900 U.S. versus the causes of death in 2010 in the US. In the 1900s, people were dying. 53% of the people were dying of infectious diseases, okay? Most of them children, or most of them pregnant females, or people who just gave birth to their kids, okay? Most of them were dying for infectious diseases, many different types of infectious diseases, diarrhea, respiratory illnesses, etc., okay? By 2010, we have decreased, as a species, we have decreased the deaths due to infectious diseases to only 3%, which is reasonable, okay? And of course, people were dying of other causes, people still have to die, and they were dying of other causes such as heart disease and cancer and many other things. And then scientists are working to solve cancer and to solve heart disease, and then it is possible that we're gonna be able to further prolong the life expectancy of people, okay? But these two numbers should tell you that antibiotics have been very important for human health as a whole, okay? So antibiotics are definitely essential for the global healthcare system, and we cannot live without antibiotics. There is no way in which we can go back to the situations that we were having before antibiotics were in the picture. Now, the problem is that we are losing a war. We are losing a war against antibiotic-resistant microbes, okay? And again, I'm gonna have to go back a little bit and explain to you quickly what are antibiotic-resistant microbes, even though the name is probably self-explanatory. But antibiotic-resistant microbes are going to be microbes, or bacteria in particular, who are no longer inhibited, by any of these antibiotics, that resist these antibiotics. And for that reason, they're going to be producing infections that are difficult to treat, very difficult to treat. And in some cases, they might be untreatable and people might die of these diseases. The way in which these organisms fight antibiotics is going to be done through two basic mechanisms. One of them is going to be active mechanisms, but they're also going to be passive mechanisms. Among the active mechanisms, we're going to get to see that bacteria or these microorganisms have the ability to eliminate drugs or any antibiotics from the inside of or the cells, okay? So they don't reach the concentration at which they're effective. They can also inactivate these antibiotics, or they can modify the targets of these antibiotics, the molecules on which these antibiotics are bound and they're inhibited by the antibiotics. They can be modified so the antibiotics are no longer active, okay? So these are going to be the active mechanisms. But we're also going to have passive mechanisms, and apparently the passive mechanisms are the most important ones, because turns out that most antibiotics are going to be only inhibiting bacteria that are multiplying, okay? So what bacteria do, what these microorganisms do, is they just simply stop multiplying. They enter dormant stages, or they're going to be producing physical coatings around themselves, which are going to be called biofilms. And in these dormant stages, and in these biofilms, basically they stop dividing, and they really don't care about the antibiotics that are going to be flowing in our body. Okay, and as soon as the 2 weeks of antibiotic treatment have passed, then these microbes simply are gonna reawake, and they're gonna continue to produce the infection. Okay, so those are gonna be some of the main problems that we're facing with the current antibiotics that we have right now. So how is it that these antibiotic resistance appear? Well, we can blame that to Charles Darwin and his theory of evolution through natural selection. This theory, this fact of evolution, or this theory has been able to explain, or Charles Darwin has been able to explain many different things about how is it that things evolve on this planet. Among them, for example, the shapes and sizes of the beaks of these finches that are adapted to their diets. But also this theory of evolution through natural selection can explain and explains how is it that antibiotic resistance actually appears in antibiotic-resistant bacteria. Okay, so through natural selection, we're going to get to see that the original population, probably the type of population that Alexander Fleming was working on, in which most of the bacteria, most of the microorganisms were susceptible to the antibiotics, but there were a couple of mutants in that population, and these were random mutants, completely random mutants in this population that were slightly resistant to these antibiotics, okay, or to antibiotics in general. And when these antibiotics were started to get into the picture, what we will get to see is that most of the population of antibiotic-sensitive bacteria will disappear, which is what antibiotics do, okay? But only the type of bacteria that is resistant to the antibiotics will be able to survive, okay? To survive and to duplicate or multiply over time. And over time, what we're gonna get to see is that the population, the genetics of the population have changed, in which most of the bacteria now will become resistant to antibiotics. Okay, so antibiotic resistance is basically driven by evolution, and is probably unavoidable. Okay, antibiotic resistance is probably we have to deal with evolution if we want to take care of antibiotic resistance. Okay, but turns out that the case of antibiotic resistance is even worse, because evolution has a warp drive. Okay, I don't know if anybody here remembers Star Trek or Star Wars, or you know, one of these fiction series. And then you guys remember that you push a button, you're flying on in space, and you push a button, and your ship suddenly goes faster. Okay, way faster. Okay, well, it turns out that evolution actually has a warp drive, and that warp drive is actually called horizontal gene transfer. Okay. And in this horizontal genes transfer, what we're going to get to see is that this genetic information, these traits that, for example, we pass to our children directly by, for example, in this case, by multiplication. In the case of bacteria, they're going to be, bacteria can take genetic information from other bacteria that are not directly related to them, and then they can incorporate this genetic information into their own genes or into their own cells. Okay. This type of horizontal gene transfer has been widely reported in the literature. For example, we know of many different cases in which antibiotic-resistant genes travel from E. coli to Streptococcus pneumonia, which are completely different bacteria. They're separated by billions of years of evolution, and suddenly we're going to get to see that all of these genes involved in antibiotic resistance are going to be spread like wildfire to many different types of bacteria. And in all of these types of bacteria, they're going to continue to evolve, and better versions of these genes are going to appear, and they're going to be again spread to many other types of bacteria, probably back to E. coli, and then, you know, making a very complicated evolutionary situation. Very unpredictable, with very unpredictable problems. Okay, so so I have I have probably explained to you guys, why is it that we have antibiotic resistance, but I now would like to talk to you guys about the bad guys, okay? So in order for me to talk to you guys about the bad guys, then let's use this a little bit of analogy. Probably some of you guys remember the Magnificent Seven movie. It's a good movie, I guess. There was a redo of the movie a few years ago. It was not really great. But, thinking about the Magnificent Seven, I'm going to be talking to you guys about the Hateful Six, okay? These are six of the worst guys, but these are not only six, okay? We have 18 microbes on the list of priorities, okay, to produce new or to obtain new therapies. But I'm going to be talking to you guys only about the worst six, okay? We're going to have Enterococcus bacteria, Acinetobacter bacteria, Staphylococcus, 2 types of Enterobacteria, and Pseudomonas. Don't worry, there is not going to be a quiz about it, but you probably want to keep these in mind, okay? So these, let's see how bad these guys are, okay? So these different types of microorganisms can be further classified, okay? There are different strains, different types of these types of organisms that are MDR, XDR, or PDR, okay? So the MDR type of microorganisms, and then all of these microorganisms, you have strains that are MDR, are multi-drug resistant, okay? Which means that they're gonna be resistant to at least 2 of the different types of antibiotics that we have, okay? Then we're gonna have the XDR, the extensively drug resistant, that are going to be resistant to most of antibiotics, okay? And then we're gonna have the very, very worst ones, which are the PDR, the pan-drug resistant. And these pan-drug resistant are resistant to all drugs that we have, or to all antibiotics that we have in the clinic right now, okay? In the pharmacies right now, we cannot treat these infections. These are untreatable infections, okay? And actually, there are going to be some types of bacteria, in particular Pseudomonas aeruginosa, that we work in my lab, that are resistant to even Clorox, okay? So there is no way you cannot kill these guys, okay? So these infections are really, really untreatable, okay? So these infections are bad. Okay, let's put some numbers to try to understand how bad they are. Okay, this is going to be, in this plot, you're seeing the 30-day mortality in the case that a 60-year-old person or older catches one of these infections. Okay, what is the chance of a person like this? And then 65 years is like, the range of my mom, okay, or me in a few years, okay, or some of us in a few years, okay. So what is the chances that anybody in these older than 65 gets an infection and dies from this infection, okay? Well, the numbers are really appalling. You have a 1 in 10 chance or 1 in 4 chances of dying from this infection if you catch it, okay? And it is not that hard to catch one of these infections, but it's relatively simple to die from these infections, okay? What would be the impact in big numbers? Okay, well, 3 million infections per year. Okay, not all of them are 65 years or older. Some of them are younger, but at least 3 million infections per year in the US alone. 35,000 deaths. In the case that you catch one of these infections, in particular, if you are 65 years old, you're gonna get your medical bill is gonna come at a rate of $10,000 to $60,000 per infection per patient. Okay, so who among us, and I don't think that too many of us, can afford a $60,000 bill? Okay, medical bills. We're talking about a loan. Okay, you need to take a loan in order to pay your medical bills. Is that the situation that we're facing? All of this adds up to $20 billion in direct medical bills per year, and $30 billion in lost productivity per year in the US alone. Okay, so this is the situation in the US. In the world, the situation is even worse. These are a few kind of random numbers that I wanted to show you. In the US, we have 30,000 people dying from these infections. In the world, we have 700,000 people dying from these infections. For example, 58,000 newborns dying in India per year as a result of drug-resistant infections. We have 500,000 new cases of multi-drug resistant tuberculosis per year. The next number is really, really appalling to me. We have 874,000 adjusted live years lost due to antibiotic-resistant infections in the European Union per year. Okay, that number is big. 7 out of 8 people who travel overseas, including me and many of us who travel on business, are bringing back multi-drug resistant infections. And it is predicted by 2050, the cost of antibiotic resistant infection is gonna cost us, it's gonna cost the world $100 trillion in a loss of $100 trillion. This is comparable to the cost of global change, global climate change. Okay, so we're really talking about a very serious situation. Although most of the people affected by antibiotic-resistant infections in the US are 65 years or older, in the world, outside of America, most of the people who are affected by these infections are going to be children. We are seeing antibiotic-resistant infections all over the place, and the rate goes from 30% to 90%, depending on the situation and depending on the country and depending on many different factors, okay? But these antibiotic-resistant infections are, as you guys can see, everywhere. Okay, so what is the future? What is going to be the future for us? How is it, what is it that we're doing? You know, how are we doing? What are the predictions? Are we going to be able to control these antibiotic-resistant infections? Well, the short answer is no, okay? So these are going to be the deaths that have been predicted using a very conservative method to make these predictions. We're going to be losing around 10 million people due to antibiotic-resistant infections by 2050. Okay, more than cancer, more than all the other causes of disease that I'm showing here. Okay, so this is a very big number. Okay. And then, are we facing a back to the future situation? So if you guys remember what I was telling you is that in the 1900s, 50% of the people were dying from infectious diseases, and in 2010, only 3% of us were dying of infectious diseases, right? Okay, by 2050, we're expecting to say that 60% of the people, 60-something% of the people are going to be dying again of infectious diseases. Is this a back to the future situation? So this problem is really serious, okay? And then one of the problems of these, one of the main problems of this situation is that all of these antibiotics that we have, as I have mentioned now, are not effective. Many of them are not effective anymore, and like I said, there are going to be bacteria that are resistant to all of the antibiotics that we have already discovered. And we have a big antibiotic discovery void, okay? We are not working on solving the problem of new antibiotics, okay? One of the problems is that to develop a new antibiotic, you guys can see my graph in the bottom right here. The private industry needs to sink close to $500 million to develop a new product. And they might recover their investment in 25 years. And as you guys can see, the recovery is not threefold or twofold, but it's marginal. So that's why many companies are not investing money or resources into developing new antibiotics. But the situation is that we cannot live without antibiotics. We need antibiotics. So somebody needs to do something about that. And that's why you guys have people like me and like my students who are scientists. We are really interested in solving these problems, okay? And then we need to find better antibiotics. These antibiotics that we discovered before, first of all, they were the low-hanging fruit, okay? That is the first thing that gets picked, the low-hanging fruit, okay? But these antibiotics actually were not designed. We discovered them, okay? We discovered the ones that were sitting there, and we didn't care about the mechanisms. Of course, we now know the mechanisms, but originally we didn't care about the mechanisms. We didn't care if there was a chance that we will develop quickly antibiotic resistance. So basically, these things were just picked because they were just there to be picked, okay? So part of the solution will be to design antibiotics, and I will be discussing that later today. Okay, so again, are we ready for the next pandemic? Are we ready to face these antibiotic-resistant infections with old and obsolete antibiotics? And then we are not facing one infection, okay? It is not going to be the case of COVID, right? We just have the one virus, okay? And the one virus created havoc. We're facing many different types of, at least 6 organisms that are producing all of these situations, and the problem is going to get even worse. So this illustration, I guess, that can be adapted a little bit for me to better explain to you guys how much problem we have in the current situation. And instead of just having this Kanagawa wave crushing this little boat, I think that the next pandemic is going to feel like, you know, the Kanagawa wave is about to crush the boats, but the Kanagawa wave is actually produced by Godzilla, who is about to attack Tokyo or whatever. Okay, so I'm hoping that I was able to grab your attention and to see that this is a very important problem that we as a society have to solve. Now, the question is, how did we get here? How did we get here, okay? The CDC is gonna be telling us that there are going to be something that the patient can do, that the most of these situations could have been prevented by, the main problems could have been through medical uses, okay? So probably antibiotics are, and definitely they are overprescribed. Definitely we have problems with self-medication, and definitely we have problems with a lack of patient awareness, okay? Patients don't know, people don't know how to use antibiotics, they don't know, you know, the problems that antibiotic use can produce, okay? But now I would think that the main problem actually for this antibiotic crisis and for many problems in the world is going to be greed, okay? There are concerns of the way in which we are using antibiotics for the non-medical uses of antibiotics. 70% of the antibiotics that we're producing in the world are going to be used by animals, and 30%, only 30% is going to be used by humans. The numbers actually could be 90 to 10, okay? But let's be conservative and let's say 70 to 30. And then turns out that many factories are going to be using antibiotics, not because the cattle or the chickens or whatever are sick, but because antibiotics for different reasons are actually growth promoters, which means that if with a modest investment of antibiotics, these factories can produce 30 to 40% more meat, more animal meat, if they use antibiotics. Okay, I think that is terrible. Also, we have a problem with water cleaning facilities. Our water cleaning facilities don't have the technology to eliminate antibiotics from the water. So there are many studies that are showing, for instance, that after water is being recycled and dumped back into Michigan Lake, we're talking about Chicago, okay? After that, after the water has been processed and sent back to Michigan Lake, we can definitely measure antibiotics in that general area in the North Shore of Chicago, where this water processing plant is operational. So we need to invest, we need to do something about water cleaning technologies, and we need to eliminate antibiotics as much as we can from the water, because what is going to happen is that all of these antibiotics are going to be released into the environment. The bacteria that are happily, without messing with anybody, without bothering anybody, in the lake are going to be producing their own mechanisms to avoid, to escape antibiotic treatment. And these genes are going to find a way to get into humans and produce problems for us. Okay, we need to find a way to eliminate antibiotics from residual water. And this picture just tells you how convoluted and how difficult the situation is, because every time that we take antibiotics, there is a chance that we're going to get that George here is going to be getting some part of the bacterial microbiota to become resistant to these antibiotics. They're going to be transmitted either to the family by direct contact, or if they go to the healthcare facility, they're going to be transmitted to other patients or if these antibiotics are going to be used to as growth promoters or going to be used in farms, what we're going to get to see is that the meat that we're going to be eating is going to contain both antibiotics and antibiotic-resistant bacteria, and the water that is coming out of these facilities is going to be used to irrigate or fertilize or whatever our crops. And again, this is creating a big environmental problem that at the end of the day is going to hurt. Well, it's going to hurt the planet, but it's also going to hurt our pockets. Okay. Okay. So what is it that we can do about it? I think that everybody should get informed, and I think it is our responsibility to be informed about the situation. I think that if it is your choice, you should consider vegan options. And what I'm telling you right now is 70% of the problem actually is caused by the meat industry. This is something that we can do today. We can buy, or if we can afford it, we can invest in humanely raised and antibiotic-free products. These are some examples that I do. I actually I walk the walk, and I actually buy these products for about 10% extra of the price what you will get from Purdue Farms or whatever, one of these evil companies. I will actually go and see these brands, and the quality is actually better. Anyway, we need to invest in water treatment facilities. And then I'm hoping that I'm not offending anybody. We definitely need to send our physicians back to school, and they need to learn and relearn all of these different things. And they need to really understand when is it that they should be using antibiotics. And probably one of the important things that we need to do is they should be using all the antibiotics to treat infection. And if these antibiotics are not effective, then you can go to the second generation or third generation, the fifth generation cephalosporin, instead of starting from the fifth generation. Okay, well, that's another question. I think that we need to choose wisely our representatives. And one of the things that our alumni can do is you guys can invest in science, okay? You can invest in science and then help us improve the quality of education and get more students interested in sciences. We can, this is something that I will be saying to students when we definitely need more healthcare professionals. Okay, definitely don't take it the wrong way, but we need many more scientists. Okay, scientists are going to be these guys. For instance, my students, for instance, are going to be the ones that are going to be solving this problem for us. Okay, so one of the things that I will be saying is that a doctor can cure a person, but a scientist can cure a disease. Okay, I think that we really need to invest in science. Okay, I'm going to be talking now a little bit about the things that we do in my lab to see, to show you guys how is it that we're trying to help with this situation. So in my lab, we're going to be developing antibiotics, but we're not going to be just discovering. Okay, we're not going to be just obtaining the long-hanging fruit and then we're not going to be running with that. No, what we're doing is actually designing antibiotics against bacteria, and these antibiotics in general it will be more difficult for bacteria to develop antibiotic resistance. Okay, so this is how we do this. This is rational antibiotic design. Okay, so what we're going to be doing is we're going to be studying the metabolic strategies of bacteria. We want to understand how is it that these bacteria can obtain energy and how is it that they survive. Okay, we need to find specific steps that these bacteria require in order to survive. Okay, and if we find them, then we can attack them. Okay, then once that we have identified one of these steps, then we're going to be doing the structural biology, which means we're going to be studying this particular step, this particular protein to death until we have understood everything about this particular process. Okay, once that we have the structural biology, the structure of these molecules investigated and understood, and now that we have enough information, then we can design drugs using medicinal chemistry to target these specific essential steps in bacteria, without which, and if we inhibited them, then the bacteria would die. In other words, what we're trying to find is going to be the heart of the bacteria, okay, or the metabolic engine of the bacteria. If we shut the engine, or if we shut the heart, the bacteria will die, and the chances of producing antibiotic resistance for this bacteria to evolve antibiotic resistance will not be zero, but will be very slim. So we have been doing, me and my students and many other people have been working on this field for a little bit. We have been working with many different types of bacteria, in particular now with antibiotic resistant bacteria. You guys have seen in this list, you guys can see several numbers that you probably can't remember now. And in all of these, with all of these, we have done studies over the last few years, and we have found a specific enzyme. It doesn't matter what it does. It doesn't matter what is the structure, not at least not for this talk. We call it NQR. And actually, this NQR molecule is the heart of the metabolic engine of the cell. Okay, this enzyme energizes the membrane, basically makes everything run, and makes everything run by energizing, like I said, the membrane, and this allows the microbes to capture nutrients, to produce virulence factor, to eliminate drugs, to regulate the pH, to synthesize ATP, to synthesize energy. So we have found the heart of the cell and the metabolic engine of the cell, and then we can target it, okay? And but before we target it, so what we decided to do, what we have finished doing actually, is understanding the structure, studying the structure. We're using cutting edge technologies, structural biology technologies, computational analysis technologies to understand and to characterize the structure of this protein. And this structure is found in this little figure, okay? By understanding this structure, what we have found is two motifs, two parts of the protein that are not found in any human enzyme. And this is very important, okay? Because they're not found in any human enzyme, so we can make drugs that only fit in these two sites that will inhibit this specific enzyme, but will not be able to be bound to any human enzyme, and for that reason, they will not have any toxicity. That's the general idea, okay? So we have already been able to do that, to design 4 novel antibiotic classes. Turns out that these antibiotics also have antipsychotic, antimalarial, and antiparasitic activities, which is another part of the conversation, but I cannot discuss that with you guys today. But what we have found is that these novel drugs that we have been able to develop can inhibit the growth of Vibrio cholera. We work with many different types of pathogens, but Vibrio cholera is going to be one of them. Vibrio cholera produces the cholera disease, which is a very difficult to treat type of diarrhea. And then what I'm showing you here, I don't want to bore you with all the scientific details, but if we were to see the growth of the bacteria with no inhibitor, we're going to get to see these black line. Okay, the bacteria is going to grow. Okay, but if we expose them to these inhibitors, these new inhibitors, basically the bacteria stop growing, which is a definition of the antibiotic. Okay, not only they're going to stop growing. Okay, because we're shutting down the engine of the cell. But also, these cells that are exposed to the inhibitors are going to be producing internally poisons, okay? And these poisons will also kill the cell, okay? So again, the ability of these cells to survive, and if they cannot survive, they cannot evolve, the treatment of these drugs will be very, very small, okay? So imagine that you basically shut a car engine, but the engine is still running. Okay, the wheels are not turning, but the engine is still running. So this is going to create a fire inside of the car, and the car is going to vertically burn. Right? This is kind of exactly what is happening in this case. Okay, the engine is shut, but the wheels are not running, but the engine is not completely shut down. So this is producing poisons they're going to be killing these bacteria. Okay. Turns out that these antibiotics that we are producing have broad-spectrum activity, which is what we want. We just don't want to get a single drug against Vibrio cholera. We want them to have, as much that can be used with as many bacteria as we can. So, for example, we have the case of another pathogen that we're working in the lab, which is Chlamydia trachomatis, which produces chlamydia, which is a sexually transmitted disease. And I'm showing you here control experiment in which you guys can see these bubbles, these blobs. This is where these chlamydia cells are hiding inside of our cells. And after antibiotic treatment, this chlamydial infection is gone. Okay. But also, this is super exciting because I have talked to you guys about these dormant stages, which are kind of the main ways in which bacteria escape antibiotic treatment, right? So one of them is going to be this biofilm that under the microscope is going to be looking like your lawn, the lawn at home, you know, the one that you cut the weekends or whatever. Okay, so this is going to be a picture of that. And these Pseudomonas aeruginosa, which is one of the very bad guys one of the hateful six that I mentioned before. If we were to treat this escaping form, this antibiotic escaping form, with these new antibiotics that we have obtained, basically the biofilm is gone. Okay, so this is great news. We have also tested these with other dormant stages also in chlamydia trachomatis. And the only thing that you need to keep track of is the intensity of this band over here. I'm hoping that you guys can see my pointer. But in this tryptase band, you guys can see the concentration of dormant stages. And then after the inhibitor treatment, the band is pretty much gone. So we were able to kill the dormant stages also with these antibiotics. Okay. So as a quick summary, I'm about to finish. We have found a very good target that we can exploit. We have identified several compounds that have antibiotic properties. We are conducting lead optimization. I'm not showing you any of that data today, but we are doing lead optimization, and we have improved the potency in only 3 steps. We have already improved the potency of these drugs by tenfold. And then we are also we're going to be conducting preclinical trials soon, okay? And the last thing that I wanted to mention to you guys today is that using the same strategy that I have described to you guys before, we can also produce antivirals, okay? We have a patent pending for a COVID-19 treatment, and we're also working on developing an antiviral against this specific type of viruses, which are going to be called henipaviruses. I would like to thank all my students, but I would also like to thank the institutions or the foundations that are supporting the research in my lab, in particular the National Institutes of Health and the Seagull Foundation. And if you're interested in contributing, your name could be here too. I'm going to be recommending to you guys, if you're interested in knowing more about this antibiotic resistant as a global threat, you guys can go ahead and check this CDC webpage. And thank you very much. And you can follow us in Facebook and in Instagram. Thank you.
Thank you so much, Professor Juárez. So we have one question that was sent to me. And just a reminder for anyone else, if you have more questions, please feel free to type them in the chat. So we had a question that was submitted. Is this a worldwide problem with antibiotic resistance, or is it worse in the United States?
It is a worldwide problem. In the United States, it is producing lots of problems. The bigger problem that we have in the United States is the economic cost, okay? Because we invest in our people. We have the means to invest in our parents, right? In our older seniors, right? In the world, basically, in developing countries, the situation is that people who get these diseases just basically die, right? So, well, they have 1 in 10 or 1 in 4 chances of dying. They just basically, go with the chances, and they cannot afford the intensive care units, or they cannot, there is nothing that for them to do. So that's a terrible situation.
And we had one other question. Are there alternatives to Are there alternatives to antibiotics for some cases of disease?
Yes, there are some alternatives, but the alternatives have their issues. Okay, one of them is going to be to use. They call it antibiotic peptides. Okay, these are going to be little proteins that are produced by other types of bacteria to kill certain types of bacteria. Okay, so you can use that. You can use these components that are going to be produced by bacteria. But the problem is that these components are immunogenic. So you can use them basically 1 or 2 times. And if the infection or the administration of the peptides is not successful, then you're stuck. You don't have anything else to go to. Okay. There is going to be also phage therapy, in which we're going to be using viruses that normally kill bacteria to treat some bacterial infections. But the problem again is that these phages are going to be immunogenic, okay? Which means that as soon as our body sees them, we're going to be mounting an immunological response, okay? So yes, we can phage therapy can be used 1 or 2 times, but not more than that, because the immune system will respond immediately to them, and then you can actually have an anaphylactic shock, and then you will die of your third administration of the phages. Okay, so the antibiotics, we really need to put more work on the antibiotics, because antibiotics are not immunogenic.
Well, thank you so much. This has been incredibly interesting. I think that was all of the questions that were sent to me in the chat. So thank you again so much, and thank you so much for everyone who attended. We will be taking a break for July and August with Alumni University, and we'll be resuming again in September. So please stay tuned on our website, and we hope everyone can join us again in the fall. So Thank you again.
Thank you.